This is Dr. Tuncay Delibasi, an endocrinologist, clinical geneticist, MD. Today's topic is comparison of therapeutic characteristics of islet cell transplantation simultaneous with pancreatic mesenchymal stem cell transplantation in rats with type 1 diabetes mellitus. The abstract of the article is: the present study aimed to investigate effect of pancreatic mesenchymal stem cell transplantation simultaneous with islet cell transplantation on islet liveliness and thus on the treatment of diabetes in type 1 diabetic rats. Blood samples were obtained from the rats 30 days after transplantation, and then their livers and pancreases were kept in 10% formaldehyde and the experiment was ended. Transplantation of islet cells together with mesenchymal stem cells showed beneficial effects in terms of prolonging survival of islet grafts, suggesting that transplantation of mesenchymal stem cells together with islet cells during clinical islet transplantation may be beneficial in increasing the number of non-insulin dependent patients in type 1 diabetes. The keywords and abbreviations used in these article. To read these you can pause the video. Let's review the methods. Experimental animals. Blood glucose concentrations of the animals were measured after 72 hours, and the rats with blood glucose concentration exceeding 350 mg per deciliter were considered diabetic and included in the study. Islet cells isolation. The islet cells were transferred to T25 culture flasks under laminar flow, and 5 ml of RPMI medium was added after being passed through the 0.22 micrometers filter. After 48 hours, the islet cells were stained with fluorescein diacetate, FDA/propydium iodide, PI, for the assessment of vitality. There is more in methods. Mesenchymal stem cell isolation and immunophenotyping. Transplantation. Histological analysis. Statistical analysis. That are given these as article. The link is given the description. Let's see the results. The mean value of vitality was 86.0 plus or minus 2.2% in the islet group undergoing islet transplantation and 84.1 plus or minus 1.7% in the islet plus MSC group, with no significant difference between the groups. P equals 0.393. As shown in these figure islet viability rates, islet alone and islet plus mesenchymal stem cell. There was also a significant difference between the groups in terms of change in the mean blood glucose concentration with time, P equals 0.027. As shown in these figure. The mean blood glucose of groups. Based on histological examination of the liver, swelling in the periislet hepatocytes was minimal in the islet group and moderate in the islet plus MSC group. Vacuolization and inflammation in the islet cells were minimal in both groups. No steatosis was determined in both groups. In the received combination of islet and pancreatic MSC, insulin and glucagon in the islets of hepatic sinusoids were stained. As shown in figure. Islet plus mesenchymal stem cell grafts, insulin staining of islets in the liver. A, or glucagon staining of islets in the liver sinusoid. B. In conclusion, MSC alone were not capable of reversing streptozotocin-induced diabetes. In the islet plus MSC group, however, transplantation of islets together with MSCs was beneficial in prolonging islet graft survival. It was thought that MSC transplantation together with islet in clinical islet transplantation would contribute to enhanced proportion of insulin-independent patients in type 1 DM. So that's all for the day, and please like, subscribe and click the bell icon for